Hi everyone, Dr. Hatami is here. Welcome to our review session for the upcoming practical exam in Physiology Lab Bio 249. In this video, I'll be covering the most important concepts of the second part of our lab course to help you prepare effectively for the exam. The concepts that we are going over is about the skeletal muscle physiology, EMG, the cardiovascular system physiology, learning about the blood, heart rate, blood pressure, ECG, respiratory system, and urinary system physiology about spirometry and urinalysis. Make sure to take plenty of notes while watching this video. Remember to take notes. Promise? Okay, let's get started. The first part is about the skeletal muscle physiology, and let's start with the sarcomere. Sarcomere is the functional unit of a muscle fiber. It is made up of repeating sections of actin and myosin filament. Actin filament is also called as thin filament. And myosin filament is also called thick filament. As you remember, the actin filament is made up of three proteins. We have actin, troponin, tropomyosin, and myosin filament is composed of the myosin molecules. When we look at the sarcomere under a microscope, we see a distinct pattern of light and dark band. We can see light band, dark band, light band, and dark band, but it gives like striated appearance to our muscle fiber or muscle tissue under microscope. For the structure of the sarcomere, please make sure you know Z discs or Z lines, I band, A band, H band or H zone, and M line. Between two Z discs, we have sarcomere. So Z lines or Z disc. So let's draw a sarcomere and then label this part. So we have Z line or Z discs, which Z lines or Z disc define the boundaries of each sarcomere. So as we can see, from Z line to another Z line would be our sarcomere. And then we have the actin filament or thin filament. As you can see, a thin blue line in this image. And the actin filaments are anchored to the Z line. So this is our actin filament. Then we can find the region, the area, which it's dark. The dark area is called A band. So A band would be our dark band or dark region, which the dark band is where myosin filaments are present. Where are the myosin filaments? Here you can find these red color. It's showing our thick filaments or myosin filament. The length of the myosin filaments under microscope, you can find a dark region. So here we have the A band or dark band, which is composed of the myosin filament, or the length of the myosin filament would be A band. And then the light area, which you can find here at both sides of the Z line, which is the area contains only actin filament, would be our I band. So we have I band here, I band or light band, which it composed of only thin or actin filament. Next part of the sarcomere, which is important to know, is the H zone. The H zone is the central part of the A band where there is no actin and we have only myosin. So if I want to show the H zone, let me use this color. So here, which only composed of the myosin filament, that would be our H zone or H band. So here we have H zone or H band. And the M line is located in the middle of the H zone. It holds the myosin filaments in place. Okay, so let's review. So for the sarcomere, which is the functional unit of the muscle fiber, we have to know the sarcomere structure, Z disc, I band, A band, H band, and M line. As you remember, when we are looking at the sarcomere under microscope or muscle fiber under microscope, we can see the light and dark regions or striated appearance. The light band is also called the I band, and the dark band is also called the A band. So then we learned that the actin filament or thin filament is going to be connected to the Z line directly, and between the actin filament we have the thick filament. So then the region that here, which only composed of the actin filament, is called I band. So please remember the I band. Please take notes. I band, it's our light area which composed of only thin or actin filament. Okay, let's get back to our image. Here, then we talked about the A band or the dark area, which it composed of the myosin filament. That would be the length of the myosin filament. So then our A band, which would be our dark band or dark area under microscope, it is the length of the myosin or thick filament. Okay. So as you can see here, where we have the A band, there are some regions we have overlap between thin and thick filament, and there is an area which only myosin filament gonna be found. There is no actin, we have only myosin filament. That is called H zone. So please take note that we have the H zone, which the H zone is the light area, the central part. So the light area at the central part of A band, which is actually composed of only myosin. Only myosin filament gonna be found here. There is no actin. Okay, so let's talk about the sliding filament theory. The sliding filament theory explains how muscles contract. During contraction, the actin filaments slide over the myosin filaments. The sarcomere shortens, but the length of the actin and myosin filaments remain constant. This sliding is driven by the interaction of myosin heads binding to actin, forming cross bridges and pulling the actin filaments inward. And now we are going through the steps of muscle contraction involving myosin and actin. So we have the attached state. So as you can see here, the myosin heads bind an actin molecule. We call this as attached state. And then the next state or step would be released state when there is detachment between the actin and myosin. Then the next step, so that is our release state. Next, we have cocked state, which during the cocked state, as you can see here, the angle between the head and tail increases. And then next we have cross bridge state, which at this state, the myosin going to be connected to the actin molecule. Then we have power stroke state, which during this state, there is another change in the myosin head shape, and this shape change causes the power stroke, sliding the filaments past each other and shortening the sarcomere. And then we get back to the another like cycle. So let's see what is the causes for each state. The myosin and actin are attached during the attached state, right? And then we have detachment state or release state, where as you can see in this picture, there is no connection or binding between the actin and myosin anymore. So whenever the ATP molecules bind to the myosin head, detachment between the myosin and actin is gonna happen. So when you see there is no attachment between actin and myosin, and then you can find the ATP molecule bound to the myosin head, that would be a released state. Then the next state, which is cocked state, as you can see, the ATP has been hydrolyzed, and now we have ADP and PI connected to the myosin head. So the ATP is hydrolyzed into ADP and PI. This will cause the myosin head to change shape to the cocked state or cock into position, which is because of the when the angle between the head and tail of the myosin molecule increases. The next step, we have the cross bridge state, when there is physical connection between the actin and myosin is going to happen. And then the next step, which is the power stroke state, when PI is released from the myosin head, it causes another change in shape. This change shape causes a power stroke, which makes sliding the filaments past each other and shortening the sarcomere. And then when the ATP is released from the myosin head, this step will repeat until the signal for contraction is removed. So make sure you know during the release state, we have the ATP molecule involved by binding to the myosin head. During the cocked state, we have hydrolyzing of ATP. So ATP, it's going to be hydrolyzed. And then you see ADP and P here, PI here. And then when we have the power stroke state, you can see the PI has been released. Next, let's talk about the twitch, summation, unfused tetanus, and fused tetanus, which there are different ways to produce tension. A twitch that you can see a graph of twitch here is when a single action potential causes contraction and full relaxation. So when we have a single stimulus, then a single action potential or single stimulus is causes the contraction and relaxation. That is called a single twitch. So one stimulus, we have full contraction and full relaxation. Summation is when we have multiple action potential or stimuli. So then when the frequency of action potential is increased, we have contraction, but instead of full relaxation, we have partial relaxation. You can see the graph of summation or wave summation or temporal summation here. You can see the stimulus. So when we have the stimulus, we have contraction. But instead of having fully relaxation, we have partial relaxation. So something like that. Stimulus, we have contraction, partial relaxation. Another stimulus, contraction, and then partial relaxation. Another stimulus, contraction, and partial relaxation. Then for incomplete tetanus, when the frequency or action potential or stimuli increases even more, the summation is reaching to the maximal tension. So then it's called incomplete tetanus, but as you can see for incomplete tetanus, still we do not see the full relaxation or no relaxation. Still we see partial relaxation. So then you can see that by increasing the stimulus or frequency of the action potential, our tension gets to the maximal range or the maximal tension. We have contraction and partial relaxation, another contraction and partial relaxation. That would be our incomplete tetanus. For complete tetanus, the tension gets to the maximal force, but we do not see any relaxation. There is no relaxation at all. So please make sure you know for a twitch, we have a single stimulus or a single action potential. We are going to see a full contraction and a full relaxation. Then when the stimuli is increasing, then we can find relaxation, but contraction, but partial relaxation between these stimuli. And then as you can see, our force does not get the maximal tension. For the incomplete tetanus, when frequency even increases more, then we may reach to the maximal tension, but still there is a partial relaxation, which it's showing the shaky muscle contraction. For fused tetanus, we do not see the relaxation at all. There is no relaxation. So no relaxation. For the fused tetanus, we have partial relaxation. For both summation and incomplete tetanus. And for twitch, there is full relaxation. And as you can see, the twitch force does not get to the maximal tension, and also for summation, but for incomplete tetanus and complete tetanus, the force gets to the maximal tension. Okay, next concept is about the blood physiology. Blood is made up of several components. We have red blood cells, or erythrocytes, white blood cells or leukocytes, platelets, which we call them as formed elements, and then the liquid part of the blood is called plasma. To study these components, we use a process called centrifugation. The spinning separates the blood into layers based on density. So as you can see, the hematocrit tube that we did in our lab, after centrifugation, we could separate our blood into some layers. The bottom layer that you can see with the red color, it contains the red blood cells, which are the heaviest part of our blood. Then the middle thin layer, which is like white color, is called buffy coat. Contains white blood cells and platelets. And the top layer is the plasma, which is mostly water and proteins. So as you can see in this picture, after centrifugation, we could separate our blood to the erythrocyte, the red part on the bottom, the yellow part, plasma on top, and between the plasma and erythrocyte or red part, we find out the buffy coat, a very thin layer composed of the white blood cells and platelets. Erythrocyte going to be 45% of our blood normally, and plasma is 55%, and less than 1% would be the buffy coat. Now let's talk about the hematocrit. The hematocrit is the percentage of your blood that is made of the red blood cells. Okay, so then you should be able to label these blood components after centrifugation. We have RBCs, or we can call this region as hematocrit, because this part, it's showing the amount of red blood cell we have in our whole blood, or the percentage of the red blood cells in the whole blood is called hematocrit. And then the top part, which you can see like a very light yellow color, would be plasma, the top part. And then in the middle between these two, we have buffy coat. Okay. And then it's important to read the hematocrit. We use the microhematocrit capillary tube reader to read the hematocrit. It's important to place our hematocrit tube on the chart properly. Make sure that the bottom of the blood, which it was red color, touch the 0% line. So that is the bottom of our blood, the red part. It has to touch the 0% line, and then you have to slide the tube along the chart till the top of the blood, which was our yellow part, the top part of our plasma, it should reaches the 100% line. Now go ahead and read the top part of the blood, red blood cells here, the red column on the scale on the right side, and that would be the hematocrit number that you need. Or another review, you can see here the bottom part of our blood, the red part, is going to touch the 0 line. Then the top part of our blood plasma should touch the 100 line, and then and this is going to be our reading point that we have to find the correct number on the right side for the hematocrit. Now let's practice. On this picture, you can see two examples. Let's see what would be the hematocrit for each of these. I'm going to give you a minute to find out. Okay, so as you can see here, the bottom part of our blood is touching the 0 line, which is correct. We have to make sure our the placing is correct and accurate. And then the top part of our blood, which is plasma, and you can see by the yellow color, it touching the 100% line. And then that would be our reading point. The 20% would be the hematocrit for this example. How about this one? Let's make sure if the placing is correct. Does the bottom of our blood touching the 0? No. This is the bottom part of our blood, not this one. Okay, the bottom part of our blood should touch the 0 line here. And does the top part of our blood, which is plasma, is touching the 100 line? That's not correct, right? So it means that this placement is not correct and we cannot actually measure or read the accurate hematocrit number for this example. Next concept, which is important, is the hemoglobin concentration, which we use hemoglobinometer to measure the hemoglobin concentration. If you remember, the hemoglobin is an iron-containing protein that is found inside the red blood cells, and its main function is to carry oxygen. So the hemoglobin content is indicator for oxygen transport. Next concept is MCHC. MCHC is also called mean corpuscular hemoglobin concentration, which is the average concentration of the hemoglobin in a given volume of hematocrit. You should be able to calculate the MCHC, but don't be worried about the formula. We are going to provide you with the equation of MCHC. You don't need to memorize it. So the MCHC equals to hemoglobin concentration multiplied by 100 divided by hematocrit. And then the MCHC unit is percentage. And you only need these two numbers, the concentration of the hemoglobin and hematocrit concentration, which we are going to give you this number as well. The only thing that you have to do is to put this number, hemoglobin concentration is 12, multiply by 100, and then 36%, which the percent would be the unit for our hematocrit. So we have 36. Then you are going to calculate the MCHC for this example. You can use a calculator to calculate this, which the MCHC for this example would be 33%. The next calculation that you need to do for the blood physiology is MCV. MCV means mean corpuscular volume, which is the factor measuring or showing the red blood cell size. For the MCV also, we are going to give you the formula, the equation, which is going to be the hematocrit. Percentage would be the unit for hematocrit multiplied by 10 divided by RBC count, which the RBC, the unit for RBC is million per microliter. So then you only need to have these two numbers, the hematocrit and the RBC count. So for the hematocrit concentration in this example, we have 42%, and for RBC count, we have 5.7 times million RBCs per microliter. And then the way that we are going to put this number in our equation is that the hematocrit 42% would be the unit multiplied by 10, and then the RBC count is 5.7, again million per microliter would be our unit. Okay, you're not going to multiply it by million. Okay, please don't do it. We are going to consider the million as our unit. So the only thing that you should do is 42 multiplied by 10 divided by 5.7. Let's see what's going to be our number. So the number that you can get for the MCV is 74. Next, we have blood typing test. Make sure we have 8 blood type A positive, B positive, AB positive, O positive, and A negative, B negative, AB negative, and O negative. One of the tests that we can do in lab to figure out what is our blood type is to use the antibody by adding to the blood in the lab and see if we can get a reaction or agglutination or clotting. That means that the antibody and proper antigen are bounded right now. So here you can see that we use the anti-A or antibody A, anti-B, antibody B, anti-D, and this is our control group. This means that, oh, there is no reaction. So this image is showing there is no reaction when we do not adding anything to our blood, okay? But then you can see some reactions after adding the antibody to our blood. So this means we got reaction. What does it mean after adding anti-A to our blood and got reaction? It means anti-A bind to the antigen A. After adding anti-A, we got reaction means there is antigen A present in our blood. After adding anti-B, no reaction, so no antigen B. After adding anti-D or anti-RH factor and we got reaction, it means there is RH factor or antigen D present in the blood. So when a blood has antigen A and RH factor, what does it mean? What type of blood is that? That would be A positive. Next example, we got reaction by adding anti-A means there is antigen A present, no antigen B, no RH factor, so that means our blood type is A negative. For this example, did we get any reaction after adding anti-E? Means there is no antigen A. Do we have antigen B? Yes, because we got reaction after adding anti-B. So we have antigen B present, RH factor present. When the blood has antigen B and RH factor, the blood type would be B positive. And let's see, when we added anti-A, B, D, and we got only a reaction with anti-B means there is only antigen B present. So our blood type would be B negative. And look at here, after adding anti-A, we got reaction. So it means there is antigen A, we got reaction with anti-B, there is antigen B present. We got reaction with anti-D, there is RH factor present. So then our blood type would be AB positive. Next, we got reaction with anti-A. So we have antigen A, we have antigen B, but there is no RH factor. So the blood type is AB negative. And here we only got the reaction with anti-D. So it means RH factor is present. Our blood does not have antigen A or B. So the blood type would be O, right? And then we got reaction with anti-D. It means the RH factor or antigen D is present. So our blood type is O positive. When there is no reaction with anti-A, anti-B, anti-D, it means there is no antigen A, no antigen B, no RH factor, and our blood type is O negative. The next topic which is important to know is about the blood transfusion. Who can give blood to who and who can receive blood from who. So the easy way to remember that the O negative is our universal donor. Is the universal donor. It means the O negative can be given to anyone, but they only can receive from O negative. And then AB positive is the universal recipient. Is our universal recipient. Means that the AB positive blood can receive from all, but they only can donate to AB positive. Then type A can donate to A, and AB. So A can donate to A, obviously, and AB. Because the AB, they already know the antigen A. And then the type B can donate to B and AB. To who? They already know the antigen B. Okay? And then positive, so it means, so if we have A, you can see that here. The positive can donate to only positive, but negative can donate to both positive and negative. So it means that if your blood type is A positive, you only can donate to A positive and AB positive. If you are A negative, so means that you can donate to both A positive, A negative, AB positive, and AB negative. And also for B. If you are B positive, you can donate to B positive and AB positive. But if you are B negative, you can donate to B positive, B negative, AB positive, and AB negative. And okay, so that is the easy way to remember. Please do not memorize this. You have to understand based on the concept of the antigen and antibody, who can donate to who or who can receive from who. For receiving, there's also an easy way. The O negative only can receive from O negative. The O positive also, they are going to receive from O only, but both negative and positive. The A negative can receive obviously from O and also A. So, but if they are negative, only can receive from negative. And then the A positive, it should be O and A, both positive and negative. And B also can receive from O and B. If they are negative, only negative people can they can receive from, and if they are positive, it's okay to given them both positive or negative. How about for AB? So the AB, they can receive from AB, from A, from B, from O. If they are negative, they can only receive from negative blood. If they are positive, they can receive from both negative and positive. Next concept is about counting the number of RBC or erythrocyte. We use the hemocytometer as the device that we use to is a specialized counting chamber we use for counting the blood cells. So that is our hemocytometer. But it's also important how to count the cells in our hemocytometer. You will count all of the cells within the large quadrants on the hemocytometer. Then count all of the cells within each quadrant except those on the far right edge and lower bottom edge of the quadrant. So let's do that. For our quadrant, we are going to define the top edge, left edge, right edge, and then the bottom edge. Okay, so then you can find 3 lines. On the top and left edge, if the cells are touching the first line, middle line, and also third line, it is okay. But if they are on the other side of third line, we do not have to count them. So as you can see in this picture, this red blood cell is going to be found on the other side of our third line. So we are going to cross this out. Or also this one is going to be found on the other side of our third line. We are going to cross this out. But how about for the right and bottom edge? For the right and bottom edge, if they are touching the middle line, we're not going to count them. Okay, so we are going to cross them out if they are touching the midline. The only cells that we can count on the right and bottom, it's okay if they are touching the first line, but not the second line or the middle line. So the middle line for the right and bottom edge, we are going to cross them out. For bottom and right edge. Okay, let's practice a little bit more. So you can see here our square or quadrant, and then you can find these all red blood cells inside. We are going to count them, but be careful about the cells on these top, left, right, and bottom edge. So look at here. This one is gonna be found on the first line. They are touching the midline. This one is touching the midline and also touching the third line, which is okay. We are going to count this, but if the cell is going to be found here on the other side, touching the third line, the other side, we are not going to count them, okay? But if they are still on this side, we are going to count them. Actually, half of them going to be found on this side, we are going to count them. And also for the left edge, look at this. It's touching the midline. We're going to count it. It's touching the midline, touching the third line, but still it's going to be found here. We are going to count it. But again, if there is one touching the third line, but going to be found on the other side, we are not going to count them. But look at here. There is another story for the right and bottom edge. If they are counting the midline for the right side or like beyond the midline, we're not going to count them. Look at this. This is touching the midline. This is touching the midline, third line. We're not for sure. We're not going to count them. Okay. So it means that for the right and bottom line, if they are touching the first line, just okay. Not the midline, or obviously third line. But for the top edge and left edge, if they are still touching the third line and we can see half of the cell on this side, we are going to count them. Okay, next topic, we are going to cover the cardiovascular system important concepts. We learned taking blood pressure by using the sphygmomanometer, this device, and also stethoscope. And also we practice listening to the heart sounds by using the stethoscope. And also for the cardiovascular system lab, we learn how the ECG device is going to measure the electrical events of the heart. So please make sure you know that is the sphygmomanometer, the blood pressure cuff we use to measure the blood pressure, and also we use the stethoscope for taking or measuring the blood pressure. And for a stethoscope also we use this device to measure listen to the heart sounds, and also the ECG device is going to measure the electrical events of the heart. When you manually measure blood pressure, you wrap a cuff around the upper arm and inflate it to temporarily stop blood flow. Then slowly release the pressure while listening for the sounds of blood flowing through the arteries with a stethoscope. And then we are going to actually record the number after hearing two sounds. These sounds indicate systolic and diastolic pressure. The systolic pressure is gonna be is taken when you hear the first corticof sounds, which means the flow of the blood through the artery. So the first sound after you're going to release the pressure, the first sound, So the first Korotkoff sound, after hearing the first Korotkoff sounds, that number would be our systolic blood pressure. And then the diastolic pressure is taken when you stop hearing the Korotkoff sound. Stop hearing the Korotkoff sounds. That number would be our diastolic blood pressure. So for blood pressure, we have two numbers: systolic blood pressure, diastolic blood pressure. The systolic blood pressure is our higher number, the top number, and the diastolic blood pressure is our lower number, the bottom number. So please make sure you know when we hear the first Korotkoff sound after releasing the pressure inside of the cough, that would be our systolic blood pressure, and when we stop hearing the Korotkoff sound, that would be our diastolic blood pressure. Okay. Then let's see what is the impact of exercise on blood pressure. Exercise can impact blood pressure. When you're active, your heart pumps harder and faster, temporarily raising your blood pressure. So we expect that during the exercise, our systolic blood pressure rises. So the systolic blood pressure increases during the exercise. Because it makes sense, because our heart works harder to send the blood out to all the body parts, so then it's gonna increase the blood pressure as well. But however, regular exercise strengthens your heart and helps keep your blood vessels flexible, which can lower your blood pressure over time. But during the exercise, when you are just doing the exercises, then temporarily your blood pressure, your systolic blood pressure is increasing. And also, let's compare a fit student heart rate and their recovery time after exercise and an unfit student. A fit student usually has a lower resting heart rate. So the resting heart rate for a fit student usually is lower, and also they recover faster. Why? Because their body is used to regular physical activity. On the other hand, an unfit student might have a higher resting, they have a higher resting heart rate and take longer to recover because they are not as active. Okay? Next, you have to know how to measure the artery or how to calculate the mean arterial pressure. The mean arterial pressure is the average pressure in your arteries during one heartbeat. It's calculated by adding 1/3 of systolic blood pressure and 2/3 of diastolic blood pressure. For measuring or for calculating the mean arterial pressure, you need the systolic blood pressure and diastolic blood pressure. And also, don't worry about this equation. We are going to give you the equation, okay? But the only thing that you have to do is to make sure you know the systolic blood pressure is the higher number and diastolic blood pressure is the lowest number, and then put them inside of this equation. Let's calculate the MAP for our example. Your patient has a systolic blood pressure of 110 milligram mercury and a diastolic blood pressure of 60 milligram mercury, and then calculate their mean arterial pressure. It should be very easy. MAP equals 1/3 of blood pressure, which is 110 plus 2/3 of 60. Let's see what we are going for MAP. Another calculation which we have in our blood physiology is pulse pressure. The pulse pressure is the difference between systolic and diastolic blood pressure. It is measured in millimeters of mercury. It represents the force that the heart generates each time it contracts. The healthy pulse pressure is around 40 milligram mercury, and the equation is very simple, but you have to know this. So if the question asking to calculate the pulse pressure, you have to know that to calculate the pulse pressure you need the systolic blood pressure and diastolic blood pressure, and just get the difference between these two numbers. For example, if your patient's systolic blood pressure is 110 millimeter mercury and diastolic blood pressure is 60, just distract these two numbers, and then the pulse pressure for this specific patient is going to be 50 millimeter mercury. Next, let's see what is the impact of exercise on cardiac output, stroke volume, the heart rate. During exercise, your body requires more oxygen, more nutrients to fuel your muscle and organs, right? So, to meet this increased demand, your cardiovascular system undergoes several changes. So the heartbeat getting higher, the stroke volume getting higher, and then cardiac output also going to be increased. Make sure you know the definition for each of these: the cardiac output, the stroke volume, the heart rate. The heart rate is the number of heartbeats per minute. Normally, a healthy individual person can have like between 60 to 70 beats per minute. That would be their heart rate. And the stroke volume is the amount of blood pumped from the heart per heartbeat. And then if you multiply the stroke volume and heart rate, you are going to get the cardiac output, which cardiac output means the volume of blood pumped from the heart per minute. So make sure you know the definition of each of these concepts: cardiac output, stroke volume, and heart rate, and see what's going to be the impact of exercise on each of these factors. Obviously, when you're exercising, your heart rate increasing, and also because our body needs more blood, so the amount of blood pumped from our heart per heartbeat gonna increase. So it means our stroke volume also gonna be increased. When the stroke volume, heart rate increase, the cardiac output also would be increased. Next concept is about the ECG, or electrocardiogram. The ECG, or electrocardiogram waves, represent the electrical activity of the heart as it beats. There are several key waves. We have P wave, the first wave that we are going to record, QRS complex, and then we have T wave. So we have P, QRS, and T wave. These are the waves that we are going to record. When we are recording the P wave, what is happening in our heart is atrial depolarization. So it means the P wave is actually represent the atrial depolarization. Polarization is our P wave. During the QRS recording, our ventricle gonna be depolarized. So we have during the QRS, we have ventricular depolarization. So we have ventricular depolarization happening here. Okay, so that's going to be our QRS. Then when we are recording the P wave, ventricular repolarization is happening. Ventricular repolarization. That is our T wave. So here we have T wave. But what is missing? We got atrial depolarization during P wave, ventricular depolarization during QRS, ventricular repolarization during the P wave. What is missing is atrial repolarization. Actually, the atrial repolarization is happening at the same time as QRS, because when the ventricle gonna be depolarized or contraction of the ventricle is happening, it's making a very huge like wave that the tiny atrial repolarization wave gonna be hidden at this time. And let's see how exercise can impact the ECG waves. During the exercise, the amplitude of each wave, the amplitude, it means how tall our wave gonna be, it's gonna be increased. So how does exercise impact the wave amplitude? It's gonna be increased. How about the wave duration during the exercise? The wave duration gonna be decreased during exercise. Okay, and make sure you know the abnormal ECG. One of them is sinus bradycardia. The sinus bradycardia, you can see the regular rhythm. We can find P, Q, R, S, T, all gonna be regular. And then, but the problem is that the heart rate should be less than normal, less than 60 beat per minute. So then you can find each of these equals, like it shows a heartbeat, a cardiac cycle. So if we can find a low number of the heart beats, so the heart rate decrease during the sinus bradycardia. How about the sinus tachycardia? The sinus tachycardia is showing that we have the more heart rate. The heart rate would be more than 100 beats per minute. But still, the wave are irregular. We have PQRST, PQRST. Then we have ventricular asystole. When there is no rhythm, there is no PQRS, and the heart rate is absent. That is called ventricular asystole. Atrial fibrillation. The atrial fibrillation, as you can see, the rhythm is irregular. There is no P wave, so we cannot find a P wave, and also we can find the wavy baseline. So we have a wavy baseline, no P, but you can find QRST. So the hint is that no P wave, and also another hint is a wavy baseline. So that is showing the atrial fibrillation. For ventricular fibrillation, look at the heart rate. It's increased too much, between 300 to 600. And look at this. We cannot find any of the wave P, Q, R, S, T. It's extremely irregular. It's extremely irregular. P wave is absent. That is our ventricular fibrillation. Next, we have the respiratory system. For the respiratory system, make sure you know the device that we use for like measuring the lung volume and capacity is called vetless spirometer. Then we use this actually to measure the volume of expired air. So we call this equipment as vetless spirometer to measure the volume of expired air. Okay. Next, it's important to know how to read a vetless spirometer to measure the spirometry volumes and capacity. So as you can see, the gauge of our spirometer is starting from 0 and ending at 7,000. But the problem is that between 0 and 1,000, there is no line that we can read the number less than 1,000. So for long volumes that are expected to be more than 1,000 milliliter, we can start at 0 ml, But for the long volumes that are expected to be less than 1,000 milliliter, we are going to start at 1,000 ml. So then before each experiment, set the dial to 1,000 ml. This would be our baseline. Therefore, 1,000 milliliter is equal to 0 milliliter exhaled. For example, if after exhalation, the needle is at 1,500 ml, then how much gonna be the volume of air moved through the spirometer in this test? You should subtract 1,500 minus 1,000. That would be your the amount of air that you exhaled. 500 milliliter. Why? Because our starting point was 1,000 and the ending point was 1,500. So please pay attention to your start point and end point, and then do this. And minus start, that would be your number. Okay, but as I mentioned, for the volume that for the long volume that are expected be more than 1,000 milliliter, you can set up your starting point at 0. So one more time, your starting point is 0. Look at your starting point, and then that would be your ending point. Look at this. We have 4,000, 5,000. Between this line would be 4,500. So it means each line equals 100. So 1, 2, 3. So it's showing 4,300. Okay. So then do this. Ending point minus starting point. We have 4,300. What was our starting point? 0. So that would be your volume that you wanted to measure for the 300 milliliter. Okay. The next concept is the long volume and capacity, which you have to be very confident drawing this chart, using this chart to calculate the volume and capacity. So please practice to draw this chart, and also you should be able to label all of these parts. The first part, which is super easy to learn, is the tidal volume. So this part that I'm highlighting, it's showing the tidal volume. What is the tidal volume? Tidal volume, it means the amount of air that normally you are going to take in and take out. So just right now, you're sitting, you're listening, you're doing something, you are normally breathing. That would be the amount of air that you are breathe in and breathe out. It's tidal volume that gonna be around 500 milliliter. Okay, now after normally breathe in, if you deeply breathe in, this amount of air that I'm going to highlight is going to show the inspiratory reserve volume. So the inspiratory reserve volume is the amount of air that after normally breathe in, you're going to deeply breathe in, which the average would be around 3,100 milliliter. And then let's try to breathe out normally and then breathe out deeply. So this amount of air that you're going to breathe out deeply is called expiratory reserve volume. So the expiratory reserve volume normally would be around 1,200 milliliter. Okay. So then if you add the tidal volume, inspiratory reserve volume, expiratory reserve volume, the amount of air that normally and deeply we can breathe in and breathe out is called vital capacity. So it means, so you can find this like equation, but just by drawing this chart and learning the definition of each of these volume and capacity. So vital capacity equals TV plus IRV plus ERV. It means the amount of air that normally and deeply we are going to breathe in and breathe out. Next, there is an amount of air that always stay in our lung, which is called residual volume. And residual volume also going to be around 1200 milliliter. So if you gonna add all of this volume, all of this four volume, it means TV plus IRV plus ERV, and also the amount of air always stay in our lung, we are going to find out the total lung capacity. Or you can say the total lung capacity equals the vital capacity plus RV, because the vital capacity would be TV plus IRV plus ERV, or you can see it here. Total lung capacity equals vital capacity plus residual volume, or total lung capacity equals the add of IRV, TV, ERV, and RV. Okay, two more. Let me just do this, erase some part. And then we have the amount of air that we are going to breathe in normally and breathe in deeply. So these two would be our inspiratory capacity. So the formula we have, inspiratory capacity equals the amount of air that we normally breathe in and deeply breathe in. How about if we are going to add the amount of air that we deeply breathe out and normally breathe out and deeply breathe out? That would be our expiratory capacity, which is going to be here. So it means from here till here, the amount of air that normally and deeply breathe out would be our IC. The amount of air that we normally and deeply breathe in, that would be our IC. So we have IC and EC here. Inspiratory capacity, and then from here till here would be our EC. Okay, one more. We have another capacity, which is called functional residual capacity, that is the amount of air always stay in our lung and amount of air that we are going to deeply breathe out. So functional residual capacity, it's going to be the add of ERV and RV. So one more time, if you know how to draw this chart and label the all part of it, and also how to define each of these volume and capacity, so finding out the equation or formula also going to be easy, and you should be able to calculate any of these volume capacity we are asking in our exam. And now let's see the impact of exercise on volume and capacities. This is a chart that's showing all of the volume and capacity of the lung at rest, and then volume and capacity during exercise. Let's see which of them is going to be changed. How it's going to be TV. So if you look at this chart and compare these together, as you can see, our total volume, the amount of air that we are going to breathe in and out during exercise, obviously getting increased. So the TV would be increased during exercise. What's gonna happen for IRV? Let's look at the IRV. So as you can see, because we have the space for the air when we are going to deeply breathe in, but some of this space has been occupied by the amount of air for TV, and now there is no more space for IRV. So it means our IRV should be decreased. What's going to happen for ERV? The same, because again, the amount of air that we are going to breathe out during the TV has been increased, so there is no more space for ERV to be increased. So then that ERV going to be decreased as well. What's gonna happen for RV? Do you think that at rest and during exercise, the amount of air stay in our lungs should be changed? No. The amount of air that stay in our lung is gonna be same at rest and during exercise, so no change. IC is the amount of air that normally and deeply we breathe in when the TV has been increased, so it means our IC should be increased as well. How about the EC? Again, EC is the amount of air that we normally and deeply breathe out. So again, because our TV going to be increased, so then there should be increase in EC as well. The FRC, or the functional residual capacity, because the ERV has been decreased, but the RV is the same, so FRC should be decreased. Vital capacity does not matter. The amount of air that we can deeply and normally breathe in and breathe out during exercise and at rest should be the same. There is no change. How about the total lung capacity? The amount of air that our lung can hold it. It does not matter at rest or during exercise. It also should be the same. No change. And let's go to the urinary system. You should know that for analysis the urine test, one of the examination that we are doing is a visual examination. For the visual examination, after collecting the urine sample, that we use the urine cup, we can look at the color of the urine, clarity of the urine. We also can measure the volume of the urine we collected, and also we can assess the odor of the urine. So for visual examination, we are able to check the color, clarity, volume, and odor assessment. For example, as you can see in this picture, that it's showing the clear urine, as we can still see these words in our newspaper. But here you can see when it's cloudy, it's hard to read the words. When it's turbid as well, there is no word at all we can see. And also for the odor, the frothy urine is the sign of the protein in urine, which is a abnormal amount. It's showing that there is abnormal amount of protein in their urine. Because normally protein is not found in urine because the kidneys filter them out, waste products, while retaining substances the body needs like protein, amino acids in our body. However, when the kidney are damaged or diseased, they may allow proteins such as albumin to pass into the urine. This can cause the urine to appear frothy or foamy. Next concept, which is important when we are going to test the urine, is measuring the specific gravity. There is a digital urine refractometer, which we can use it to measure the specific gravity by adding the urine here on this hole. Then you can find in the display there is a number. So you should be able to read this number. So as you can see, there is a number 1 and point always here, and you just simply read these the 4 numbers that you can find here. So for this patient, the specific gravity would be 1.0317. That is a number that you need. But what is the specific gravity? The specific gravity actually evaluates the body's water balance. So if you are hydrated enough or you are dehydrated, and also your urine concentration. It's showing that if there are solid in your urine or is not. For example, if someone suffering from diabetes mellitus, so we can find the glucose in their urine. So we expect they have a higher specific gravity because, again, the specific gravity showing there are some solutes inside of the urine. And then the expected, so the normal ranges of the specific gravity should be like between 1.020 to 1.030. One more time, the low specific gravity, it can show the person is hydrated well if they are normal, or it can show that there is low solute in their urine. The high specific gravity, it's showing that the urine is concentrated, so they are dehydrated, so dehydration. And also it's showing that there are a high amount of solute in their urine, solute. And also we use the reagent strips to determine the urine contents. So the reagent strips, we can determine the urine contents. For example, if there are glucose, bilirubin, ketone, blood, protein, urobilinogen, nitrate, leukocytes found in the urine. And also we can measure the pH and a specific gravity by using the reagent strips. And as you remember, after using the reagent strips, we dispose them into the biohazard soft skin. Next concept is what's gonna happen if you ingest the amount of solution which has 0.9% NaCl. If you remember when we talked about the different type of solution like isotonic solution, the hypotonic solution, the hypertonic solution. The best example for hypotonic solution was water. The isotonic solution, our 0.9% NaCl or normal saline, is the example of the isotonic solution. After consuming the amount of this solution, what's gonna happen for the for in our blood and also for our urine? So if you just follow the changes of the urine color before and after ingesting the amount of normal saline, you're gonna see there is no change at the urine color. The urine volume at first it's going to be increased and then it's going to get back to the normal, and also there is no change at their specific gravity. What's going to happen after ingesting the 500 milliliter of 0.9% NaCl? The first thing that's going to happen, obviously our blood volume going to be increased. When blood volume increasing, there is a pattern that it says the blood pressure also going to be increased, and then our cardiovascular system, the kidney is working hard to make sure that we are going to return to the normal blood pressure. So one more time, because normal saline is an isotonic solution, meaning it has the same concentration of solutes as your blood plasma. So drinking this solution would increase your overall fluid volume without significantly altering the osmolarity of your body fluids. So the osmolarity not gonna change. Okay. So what is happening in our body? Because actually in our heart, in atria, we have some volume receptors. When blood volume increase, the volume receptors gonna detect these changes, and because there are some cells in our atrium which they are going to produce and release the hormone. These hormones are called natriuretic peptides like ANP. So the ANP or atrial natriuretic peptides is the hormone going to be released from our heart in response to the increasing of the blood volume and then blood pressure. Let's see how the ANP is going to help our body to get back to the normal. The ANP actually can impact impact our medulla oblongata, the part of our brain, the brain stem, and then this medulla oblongata is going to increase the sympathetic output. And then the sympathetic output is going to help decreasing the blood pressure. And also the ANP can impact our kidney, and then ask kidney to produce and release less urine. So the next impact would be decrease in renin. The renin is the hormone which can increase the blood pressure. So when there is less renin, it's helping to decrease the blood pressure as well. So, and also another one, that the kidney is also going to increase the salt and water excretion. So by these all changes increase salt and water excretion, decrease renin production, and also decrease sympathetic output. We are going to get back to the normal value by decreasing our blood pressure. What's gonna happen when we ingest salted chips? Ingesting salted chips, there is no change in volume of the blood, but osmolarity gonna change. So no change at blood volume. But what about the blood osmolarity? The blood osmolarity gonna be increased because we are consuming the salt. Okay, so then what is gonna happen? The vasopressin gonna be released by our posterior pituitary gland, and the way that the vasopressin is going to help our body to get back to the normal value is that it's going to increase the water reabsorption in the nephron, and by increasing the number of the aquaporins in the collecting ducts. So the vasopressin target organ would be the kidney. So then it goes to the collecting duct, and then it's gonna increase reabsorb of water in the nephron or back to the blood, and also it's gonna increase the number of the aquaporin, the channels which help the reabsorbing of the water. So increase water reabsorption to conserve water by increasing the number of aquaporin in the collecting duct. What is the impact of aldosterone? So whenever we have a lowering or decreasing the blood pressure, the aldosterone hormone would be released from our adrenal cortex. The aldosterone is going to increase the sodium ion reabsorption at the collecting duct. So as you can see here in this picture, the aldosterone is going to be released from the adrenal cortex, and then the target organ would be kidney. It goes to the kidney, at the collecting duct is going to increase the sodium ion reabsorption. We are going to send the sodium to the blood. Okay, so we are going to increase the sodium reabsorption, and also we are going to increase the sodium secretion. It means decrease potassium ion reabsorption. So these two gonna happen. Sodium reabsorption, it means from the filtrate the future urine to the back to the blood, and for potassium the secretion gonna be increased. It means potassium going from blood to the filtrate, or it means decreased potassium ion reabsorption at the collecting duct. And then when you're ingesting the 500 milliliter of orange juice, because in our orange juice we have citric acid, what's gonna happen to the urine pH? So then as you can see here, as a response, our urine pH is going to decrease, and then it's gonna back to increase. So we have decrease of the pH. That is our response. Ingesting the 500 milliliter of baking soda, because in our baking soda we have bicarbonate ion, so then the bicarbonate is going to increase or make our urine alkaline. So it's gonna increase the pH or make our urine alkaline. Thank you so much for watching to this video. I hope you enjoyed and you learned and took notes. Please get back to me if you have any question, and good luck.